A very good morning to everyone and Dr. Nadia. Today, me and my team are going to talk about flow cytometry. First of all, me and my team, which are me, Kiron Kang, Hadi, Aida Safina, and Zaidato. And we are going to talk more about principle and application as well as study case of flow cytometry. What is flow cytometry? Flow cytometry is actually a combination of flow, the word flow, cell and measurement, which mean by cell calculation. Okay, as we can see here, this is the schematic diagram of flow cytometry, which have the fluidis system and optic system as well as electronic system. Okay, working on the flow cytometry, which what we said just now have three main systems: fluidis, optics and electronics. The function of fluidis is to transport cell in the stream to the laser beam for integration. Optics is where the laser to illuminate the cell in the sample stream, and optical filter to direct the resulting light signal to the detector, where after that electronics is to convert the detected light signal into electronic signal that can be processed by computer. Okay, first let's go to fluidis, which is the first part of flow cytometry. As we can see here, cytometry have the hydrodynamic focusing to present one cell at a time and consisting of central channel through where the sample is ejected and enclosed by outer shell, which the hydrodynamic area we can see here, and that's where the cell is aligned in a single file. As the shell fluid moves, it creates massive drag effect on the narrowing central chamber, so all the cells can continuously flow through it, and the effect of creating a single stream of particles are called as hydrodynamic focusing. Okay, next we will go to optics. Optics is actually the light scattering fluorescent emission provide information about the cell properties, and there are two types of beam, which are forward scatter and side scatter and fluorescence. Okay, forward scatter is based on two properties, which are size and refractive index, which forward scatter intensity roughly equated to particle size, which we always use it to distinguish and between cellular debris and living cells. However, that cell will have lower forward scatter and higher side scatter than average living cells. Okay, next, fluorescent measurement taken at different wavelengths can provide quantitative and qualitative data, which always use on intracellular molecules such as DNA and cytokines, which then, when a fluorescent dye is conjugated to a monoclonal antibody, it can be used to identify a particular cell types. Besides that, the stacking pattern of each subpopulation, combined forward scatter and side scatter data can be used to identify which cells are present in the sample and their percentage. Okay, now we are going to view the system of optics. Okay, in optics, the whole system is made up majority of optical detectors, which consists of photodiodes and photomultiplier chips. Okay, the dichroic mirror's main function is to filter out light and reflect it to the respective detectors, and then the detectors will use to detect forward scatter as well as side scatter, which photodiodes is to collect and detect the forward scatter, and photomultiplier is to collect and detect the side scatter. And then all the data will be transformed and carried to the electronics. Okay, after this we will go to the last part of flow cytometer. As we can see here, the last part of flow cytometer is actually constructed by electronics, which is a signal processing site. At the electronics part, the scatter and emitted light, including forward scatter and side scatter light, can be converted into electrical pulse by optical detectors, which after that the emitted light and data will be converted into plot diagram represented by histogram or dot plots. Okay, here are the example of histogram and dot plots. Histogram main function is to quantify the intensity of single parameter, be it fluorescent or scattered, which is SSC and FSC. Okay, dot plot is a two-parameter representative of samples properties. Okay, next I'm going to show you about flow cytometric video for a better understanding for it. Flow cytometry is actually the use to count and analyze the size, shape, and even properties of individual cells within the heterogeneous population of cells. Okay. Flow cytometric data is extremely quantitative and can be specified flow in depth of flow cytometry with software programming. Once a suspension of individual cells has been prepared and flow cytometric analysis of cells and tissue, the sample is then placed on the flow cytometer. Okay. In the flow cytometer contains several components which include the sample fluids, which move the sample fluid, laser and optics which gather the lights, and detector to send the computer output, signal output. And then the data into the form can be analyzed by the researcher. Okay, once the sample has been placed into the flow cytometer, the machine will suck up the samples, and then the cytometer will lead the cell suspension through a narrowing channel, form a single file line of the cell called the integration point. Within the sample is then across the laser beam. Okay, allowing each cells within the sample to be individually analyzed. Okay, as each cell passes through the laser beam will be scattered in multiple directions. When the laser beam is in forward manner, it is called forward scatter. When the laser beam is in sideward manner, it is called a side scatter. And then the amount of forward scatter light for each cell is detected by detector on the far side of the cells from the laser. And then the forward scatter is proportional to the size of the cells. Okay, the detector converts the scatter light into a voltage pulse which directly proportional to the amount of the light. Okay, the computer converts this data into a histogram plot for the amount of scatter light. However, the amount of size scatter light is detected by detector located perpendicular to the path of the laser beam, which collect the size scatter from the cells and internal complexity of the cells. The flow cytometer converts the detected size scatter light into voltage pulse, which directly proportional to the amount of size scattered light. Okay, by analyzing everything together, the researchers able to understand the cell size and complexity of the cells. Okay, besides analyzing the cell shape and size and complexity, the flow cytometer also can detect emitted light from the cells. Okay. From excited fluorescent molecule, you have fluorescent label such as fluorescent dyes and stains. The fluorescent molecules of fluorophores are excited by the correct wavelength of the laser beam and then passes through the laser beam. After excitation, the light emitted by the fluorophore is directed to the path with the emission filter, allow the detection of multiple different fluorescent emitting light in a single cell. A flow cytometer not only can detect the presence of fluorophore, but the quantity and relative amount of a fluorophore within a cell. A flow cytometer can detect up to 4 to 5 colors at the same time, and of course it depends on the type of flow cytometers. Some of the flow cytometers can even detect up to 18 colors at the same time. And then the data presented to the researcher in the form by histogram or dot plots. The data provided furthers the power of flow cytometry analysis by enabling the researchers to analyze individual population within a. Okay, next we are going to talk about application of flow cytometry. Flow cytometry is actually modern researchers' toolbox. Why do we say so? Because flow cytometry measures multiple characteristics of individual particles flowing in a single file in a stream of fluid, which light scattering at different angles can distinguish differences in size and internal complexity, whereas light emitted from fluorescently labeled antibodies can identify a wide array of cell surface and even cytoplasmic antigens. This makes flow cytometry a powerful tool for analysis of complex population in a short period of time. There are several applications for flow cytometry, such as immunophenotyping, cell sorting, DNA content analysis, cell cycle analysis, apoptosis, and cell proliferation assay. And the most common use for flow cytometry is DNA content analysis, cell sorting, and cell cycle analysis.
of data DNA content asymmetry of white and black membrane using flow cytometry. Figure 1 and Table 1, we can see G1 peak of week 115.94 was 4.5 times more than common week 517.96. Figure 2 and Table 1, we can see G1 peak of black mulberries 163.53 was 1.2 times more than the of berry 208.77. The ratio of 2C DNA content of the black mulberry was 10.17 times of the peak, while the ratio of 1C genome of the black mulberry was 10.17 times of the peak. Unit 2C DNA content and calculation 1C genome volume of peak was more than host of the Lolola. The reason for this variant can be attributed to the fluorochromosome difference used in the study. It can be estimated that DNA content observed after DAPI staining may be expected to differ from the PI staining. It was stated that DAPI was being prepared as the fluorochromosome for the gene. Pit dye can be used to make DNA content estimate for pit and the black mulberry in the future. This is a primary study to estimate the DNA content and genome size of the black mulberry. Conclusion: the ratio of 2C DNA content and 1C genome volume of the black mulberry was 10.17 times that of the peak, although F currency is developed and Negia dicosophora.